So now we're looking at important cells of the adaptive immune system, which include our lymphocytes and antigen presenting cells. There's going to be three types of crucial cells here. Two types are lymphocytes. There's going to be the B lymphocytes or B cells and the T lymphocytes or T cells. The B lymphocytes are involved in humoral immunity, and the T lymphocytes are involved in cellular immunity. And then we have the antigen presenting cells or APCs. These particular cells do not respond to specific antigens, but they do play an essential auxiliary role in immunity, which we'll see in a bit. For the lymphocytes, they go through some stages of development from their creation in the red bone marrow to maturation and activation. Both types, the T and the B lymphocytes, do share common developmental steps, and there's 5 general steps. We're going to look at those briefly in a diagram and then move on to a bit more details. So here is our figure 21.8 showing the 5 steps. Step 1 is the origin. So here both B and T lymphocyte precursors are going to originate in the bone marrow, in the red bone marrow. And so that's modeled there for the bone marrow, right here. And then in the maturation stage, the lymphocyte precursors that are going to end up becoming T cells are going to migrate in the blood to the thymus and mature there. So they're going to go, the ones that are going to become T lymphocytes are going to go to the thymus for their maturation. The B cells are going to stay in the bone marrow and mature there. And during the maturation of lymphocytes, they're going to develop both immunocompetence, or the ability to respond to specific antigens, and they are going to also develop self-tolerance, which means that they won't be affected by the body's own antigens. Then we're going to go on to step 3. After maturation has occurred, these cells are going to move into secondary lymphoid organs. So these areas are going to be seeded. So seeding is going to occur. And here the immunocompetent, but still they're still naive lymphocytes, which means they're not activated yet, are going to leave the thymus and the bone marrow, and that's where they're going to seed the secondary lymph organs, like the lymph node, for example. And then it is in these places where the lymphocytes would encounter their antigens, and when they do, they become activated. So the idea here is once you're in circulating within the lymph or the lymph nodes, or lymph vessels, and you encounter an antigen, that's when it become activated. And then step 5, upon activation, this is where proliferation and differentiation is going to occur. So here the activated lymphocytes are going to multiply, and then they differentiate either into effector cells or memory cells. And so there's the effector or memory cells. And memory cells and effector T cells are going to circulate continuously in the blood and lymph and throughout secondary lymphoid organs. So we can see the steps here. We went over that. In the origin, the lymphocytes originate in the red bone marrow. During maturation, the lymphocytes become educated and mature in the primary lymph organs, which is either the bone marrow or the thymus, depending on if it's the B cells or the T cells. And then 2 aspects to that includes immunocompetence and self-tolerance. And for immunocompetence, lymphocytes are made to recognize only one specific antigen. And here the B and T cells are going to display only one unique type of antigen receptor on the surface when mature, which means they can bind to only one specific antigen. And then self-tolerance here, the lymphocytes must be unresponsive to the body's own antigens. So the maturation in the thymus, for example, for the T cells, there's going to be 2 aspects. There's going to be negative and positive selection here. So looking at positive selection first, right here. Here the T cells capable of recognizing the self-major histocompatibility complex proteins, which are those that can recognize those on the cells, are going to be selected for, and those that cannot are going to be destroyed by apoptosis. And so we can see that model right up here in this part of the diagram here. Here we have a sel-MHC complex and our developing T cell. And right here in this area you can see that these two regions right here are not matching completely, so there is a non-recognition there. And if that non-recognition occurs, then we have apoptosis occurring there. So the programmed cell death. So this T cell will be selected for destruction. On the other hand, if we get a good fit with the MHC complex proteins, then we do recognize self-MHC, and the cell survives and proceeds to the negative selection process. So what happens here in the negative selection, apoptosis is initiated to T cells that bind to self-antigens displayed by the self-MHC. So here we've already gone through the business of being able to recognize self-MHC proteins up here, but now we are going to see now whether or not our developing lymphocytes, T lymphocytes, actually recognize our self-antigens, and if they do, then they're going to be selected for clonal deletion and go through apoptosis, and that's modeled here. Here we already have a good fit for the MHC proteins, but then you see this real nice fit. So that antigen, that yellow molecule in this model here, has a perfect fit. So that means here this developing would-be maturing T lymphocyte recognizes and would actually react against the body's own antigens. So that would be bad, because now you'd have cells that are attacking your own body cells. So this one would then be negatively selected and go through apoptosis and die here. And so if we have a failure to recognize, and that's the case right here, where you don't see a real good fit in this model, and in this case the cell goes on to continue for maturation to become a T lymphocyte, or a naive T lymphocyte. For B cells, the process may be similar, but this is going to happen in the red bone marrow. Okay? And not much is known about this process for B cell maturation, so not much known. But only immunocompetent B cells are allowed to mature. Those that are self-reactive are going to be eliminated by apoptosis, similar to what happens with the maturing T lymphocyte cells. So they go through clonal deletion also. And then on step 3, we're going to have seeding of secondary lymphoid organs and circulation. So here we now have our immunocompetent B and T cells, but since they haven't been exposed to antigen, they're considered naive still, or naive. And they're exported from your primary lymph organs, like the bone marrow and the thymus. And there they're going to go on to seed secondary lymphoid organs like lymph nodes, spleen, etc. And it's there that they have an increased chance of encountering the antigens if they are present. If the naive lymphocytes then encounter the antigen, that's going to trigger an activation. Okay, so when they encounter this antigens, they become activated. So in this case, the lymphocyte is selected for differentiation into an active cell when the binding to that specific antigen occurs. Okay? And this is referred to as now clonal selection. So if the correct signals are present, the antigens, then the lymphocyte will complete differentiation into an active cell. The next step after the differentiation occurs, the lymphocyte now would be, the activated lymphocyte would go through divisions and proliferate and further differentiation. So we're going to now make a bunch of copies, exact copies, and we're going to refer to those as clones. Most of the clones are going to become effector cells that go on to do battle. for the infection, and a few are going to remain to become memory cells. These memory cells are going to later on be able to respond if the same antigen becomes present again within the body, and this allows for a more quick response since the cells already have the memory of that being exposed to that antigen. So both B and T memory cells and effector T cells are going to be circulating continuously. So again, this is the same diagram we started with, and you see all the 5 steps. Make sure that you are familiar with the steps in the development of your lymphocytes. As far as the lymphocytes, the receptors that individual lymphocyte develops are quite diverse. So, and it's the genes, not the antigens. Okay, so that are going to determine which foreign substances the immune system will recognize. There's a variety of immune cell receptors, and they are a result of acquired genetic knowledge of microbes. So over time, the genes have been there to produce these antigens. So there are approximately 25,000 different gene codes for up to 1 billion different types of lymphocyte antigen receptors. And so how does that huge variety of receptors come about? And that's due to gene segments of the gene being shuffled around. And as they're shuffled around, they result in many different combinations of potential receptors for these antigens. So this table summarizes overviews the B and the T lymphocytes. And so here you can see the type of responses. For the B lymphocytes, it's humoral. For the T, it's cellular. Does the cell type secrete antibodies? For the B lymphocytes, yes. For the T lymphocytes, they do not. The primary targets for B lymphocytes are extracellular pathogens, since they produce antibodies that circulate around in the fluids. And for the T lymphocytes, this is intracellular pathogens and cancer cells. So here, this is where the T cells respond to cells themselves. And then the site of origin, both are going to originate lymphocytes in the red bone marrow. And they mature in different places though. The B lymphocytes are for bone marrow where they mature, and the T are in the thymus. The types of effector cells produced after differentiation from activation, the B lymphocytes produce plasma cells. The plasma cells are going to be the ones that actively produce antibodies. And for T lymphocytes, we have a few types. We have the cytotoxic T cells, we have the helper T cells, and then we have regulatory T cells. And both do produce memory cells. And then we have the antigen presenting cells, or the APCs. Here these cells are going to take in or engulf antigens and present fragments of the antigens to the T cells for recognition. So basically they might take in like a bacteria and digest it down and then present some of the parts of that bacteria to T cells for their recognition. There's 3 major types. There are the dendritic cells, there are macrophages, and B cells. For the dendritic cells, there's an image here on the right. These are found in connective tissue and epidermis and act as mobile lookouts for the boundary tissues. They're out there looking for any possible invading pathogens and their antigens. And when those things become present, the dendritic cells can phagocytize them and take them in. And then what happens there is the dendritic cells will, after phagocytizing, will enter the lymphatic vessels and present their antigens to T cells at the lymph nodes. And these cells dendritic cells are probably the most effective antigen presenters in your body, and they represent a key link between innate and adaptive immunity. The macrophages, these are widely distributed in connective tissue and lymphoid organs, and they do present antigens to T cells as well, which not only activates the T cells but also further activates the macrophage. And here the activated macrophage is going to become a very serious phagocytic killer, being very voracious at its job. This is also going to trigger powerful inflammatory responses and recruit additional defenses with its interactions. And then the B lymphocytes, they do not activate the naive T cells when the B lymphocytes are presenting, but the presented antigens that are presented, they're going to present to the T cells, the helper T cells, and they assist in the helper T cells activation.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.